Welcome back to the deep dive.
Mm.
Today, we're doing something a little bit different. If you've been following along with us, you know we've spent a lot of time in the engine room of the cell.
We have, down in the boiler room, shoveling coal, talking about metabolism.
Exactly. It's
Yeah.
all been about thermodynamics, heat, movement, burning fuel to get power.
Very industrial, as you say.
Right.
Lots of breaking things down to get, raw energy.
But today, we are wiping the soot off our faces. We're walking up that spiral staircase, and we are entering the library.
I like that.
We're making a fundamental shift. We're moving away from the ephemeral world of energy flow, which you use and then it's gone, to the world of information, the world of storage, memory, and instruction.
It's such a crucial distinction, isn't it? Because energy, it desiccates. It does its job and turns into heat. But information is meant to persist. I mean, think about it. Every civilization in history has tried to solve this problem of persistence. We carve things in stone, we write on scrolls, we build these massive server farms out in the desert. We are obsessed with leaving a record.
But biology solved this problem billions of years ago.
It absolutely did. It built an archive that, it repairs itself, it copies itself, and it fits inside a microscopic nucleus. It's an archive that literally contains the instructions for how to read itself.
That's the mind-bending part. So that's our mission today. We are looking at nucleotides and nucleic acids. This is the molecular basis of biological information, the actual hardware of genetics.
It really is.
And to guide us, we're using our two cornerstone texts. We're pulling the deep structural rigor from Leninger's Principles of Biochemistry. That's going to give us the what and the how, the pure chemical architecture.
And we're pairing that, as always, with Harper's Illustrated Biochemistry to get the medical reality, the so what. Because this isn't just abstract chemistry on a page. These molecules, their bonds, their shapes, that's where disease begins.
So just to be clear for everyone listening, this isn't a replacement for reading the books. Think of this as a conceptual map. We want to help you see the patterns and the why behind the what, so that when you do go back to Leninger and Harper, it clicks on a deeper level.
Exactly. Use this as a guide, a scaffold. The reading is still essential.
So let's deconstruct the blueprint of life.
Yeah.
How do we get from simple, lifeless chemicals, carbon, nitrogen, phosphorus, stuff you could find in a rock, to a molecule that can carry the instructions for a human being?
And maybe more importantly, why should you, as a student or a clinician, care about the specific angle of a bond on a sugar molecule?
Yes, why?
Because that bond angle is the reason chemotherapy works. It's why some genetic diseases are so devastating. We're going to find out why ATP is like a rechargeable battery, but DNA is like a library with a very, very strict librarian.
I love that. Okay, let's start at the beginning. Section 1, the hardware. The anatomy of a nucleotide. If I want to build a strand of DNA or RNA, I need a basic building block, a brick. What is that brick?
That brick is the nucleotide. And Leninger breaks it down beautifully. It's an incredibly elegant modular system. Every single nucleotide, whether it's in your body or in a bacterium, has 3 essential components.
Okay, what are they?
First, you have a nitrogenous base.
That's the information carrier, right?
The letters. Precisely. That's your A, T, C, or G. Second, you have a pentose sugar. Think of this as the structural scaffold. It's the thing you attach the letter to.
And the third piece?
The third piece is one or more phosphate groups. This is the linker. It's the energy source. It's what allows these bricks to connect to each other.
Base, sugar, phosphate. It sounds simple enough.
It is. It's like a little modular vehicle. The sugar is the chassis. The base is the driver that gives it its identity. And the phosphate is the fuel tank and the trailer hitch all in one.
I want to zoom in on that chassis first, the sugar. Because this is where the fundamental difference between DNA and RNA actually lives, and it's something I think a lot of us, myself included, kind of gloss over.
We do. We say deoxyribonucleic acid and ribonucleic acid so often they just become sounds.
Exactly.
But the entire difference is in that one prefix, deoxy.
Meaning without oxygen.
Right. Both molecules use a pentose sugar, which just means a 5-carbon ring. In RNA, that sugar is called D-ribose. In DNA, it's 2'-deoxy-D-ribose.
Okay, so let's unpack that. The 2' tells us where the change is.
Yes, it's a specific address. We number the carbons on the sugar ring as 1', 2', 3', and so on, just distinguish them from the atoms in the base. At that 2' position on the sugar in RNA, you have a hydroxyl group, an OH.
An oxygen and a hydrogen.
Right. But in DNA, at that exact same spot, the oxygen has been removed. It's just a hydrogen atom. It is deoxy.
This seems like such a tiny, trivial detail. We're talking about one single atom in this big, complex molecule. Why does biology go to all the trouble of ripping one oxygen atom off? What's the point?
This is one of those beautiful moments where chemistry dictates destiny. That little hydroxyl group on RNA, the OCOH, at the 2' position, it's a chemical liability.
The liability.
It's reactive. That oxygen has electrons that can, under the right conditions, essentially launch a chemical attack on its own backbone.
Wait, it attacks itself.
It attacks the phosphate group of its neighbor in the chain. It's a process called alkaline hydrolysis. It breaks the phosphodiester bond. It literally causes the RNA strand to fall apart.
So RNA is chemically designed to self-destruct, in a way.
Designed is a strong word, but it's inherently less stable.
Yes.
And that fits its function perfectly. RNA is supposed to be transient. It's a memo. You write it, you read it, and then you throw it away. You don't want old messages cluttering up the cell's operations.
So its instability is a feature, not a bug.
Absolutely. But DNA is the master archive. It's the constitution. You want that to last for 100 years in a human, or, thousands of years in a mammoth bone frozen in permafrost.
And removing that one oxygen atom is what makes that possible?
By making it deoxy, evolution created a molecule that is far more resistant to that self-hydrolysis. It's chemically more inert, more robust. It's the difference between writing something on a napkin with disappearing ink versus carving it into a granite slab.
So the D in DNA isn't just a letter, it's a preservation strategy.
That's so.
That's incredible. Okay, so that's the scaffold. Now let's talk about the letters themselves, the nitrogenous bases. We've got 2 families here, purines and pyrimidines.
Right, and every student of biochemistry has struggled to keep these straight at some point.
Guilty.
Let's start with the purines. These are adenine, which we abbreviate A, and guanine, G. Structurally, these are the big ones. They have a double ring structure. It's a 6-membered ring fused to a 5-membered ring.
And the classic mnemonic here is pure as gold.
Pure as gold. Purines are A and G. It's silly, but it works every time.
And then we have the pyrimidines. That's cytosine C, thymine T, and uracil U.
And these are the smaller ones, just a single 6-membered ring.
The mnemonic for that one is CT, the pi. C, U, and T are pyrimidines.
Right. And notice that thymine is generally in DNA, while uracil takes its place in RNA. They're structurally very similar.
Harper's makes this really useful observation about the names that I found. Helps with remembering the size.
Oh, the inverse relationship. Yes. It's a great little trick. The word pyrimidine is long, has more syllables, but the molecule itself is small, just one ring.
And purine is a short name.
For the larger double ring molecule. So if you can just remember that the length of the name is opposite to the size of the molecule, you'll never mix them up.
That's genuinely helpful. Okay, speaking of nomenclature, we have to clear up one more thing before we start building. I see the term nucleoside sometimes, and then nucleotide. What's the difference?
It's all about the phosphate. A nucleoside is just the base plus the sugar. That's it.
Okay, so it has the identity, the letter, and the scaffold, but no energy, no way to connect.
Exactly. Think of the nucleoside as a car in the driveway. It has a chassis and a driver, so we know it's, say, an adenosine car, but it has no gas in the tank.
And the phosphate is the gas.
The phosphate is the gas. As soon as you add at least one phosphate group to the sugar, it becomes a nucleotide. Now it's activated. Now it has the energy to form bonds. Now it can be linked into a chain.
This seems critical because Harper's mentions that some drugs are actually administered as nucleosides.
Yes, and that's a key concept in pharmacology. If you give a patient a nucleoside analog drug, it's inert. It can't do anything on its own. It relies on the patient's own cellular enzymes, kinases, to slap those phosphates on and activate it.
So if the cell's machinery can't put the gas in the tank, the drug is useless.
Completely useless. The phosphorylation step is the activation key.
All right. So we have our bricks. We have a deoxy sugar for DNA stability. We have our 2 families of bases for information. And we have the phosphate for energy and linking. Now let's move on to the architecture. How do we actually build the chain?
This brings us to the famous sugar-phosphate backbone. When you see that classic spiral ladder model of DNA, the 2 long twisting sides of the ladder, that's the backbone.
And the connection that holds the units together is called a phosphodiester bond.
That sounds complicated.
It sounds more complicated than it is.
Oh.
Let's break down the name. Phospho, because it involves a phosphate group. Diester, because that one phosphate group forms 2 ester bonds.
2 bonds to what?
It forms one ester bond to the sugar of the nucleotide above it, and a second ester bond to the sugar of the nucleotide below it, so that one phosphate acts as a bridge, linking 2 sugars together.
And this linkage isn't random. It connects very specific points on the sugars.
Absolutely. This is where that numbering, the 1', 2', 3', becomes so vital. The bond always forms between the 5' phosphate of the incoming nucleotide and the 3' hydroxyl group of the last nucleotide in the chain.
We hear this phrase 5' to 3' constantly in biology. It's like a mantra.
It is.
It sounds like a set of coordinates, but it's more like grammar, isn't it? It gives the whole thing a direction. That's the perfect way to put it. It is the grammar of life. Because that bond is always 5' to 3', the resulting chain has a distinct polarity. The 5' end will always have a free phosphate group. The other end, the 3' end, will always have a free hydroxyl group. They are chemically different.
So you can't read it backwards and get the same meaning?
No more than you can read dog bites man backwards. Man bites dog is a completely different story. And the cellular machinery, the enzymes that read and copy this information, are built to read in only one direction, 5' to 3'. They are unidirectional.
So if you flip the strand around, the polymerase enzyme just wouldn't even know what to do with it.
It wouldn't even be able to bind. The shape would be all wrong. The directionality is absolute.
Now, Leninger brings up a really fascinating point about the physics of this backbone. It's about how it interacts with water, and apparently this property is the main driving force for the entire double helix structure.
Yes, the hydrophobic effect.
Yeah.
This is arguably more important for the shape of DNA than even the hydrogen bonds between the bases.
Really? I always thought it was the hydrogen bonds holding it all together.
The hydrogen bonds provide the specificity, the A pairing with T, the G with C, but the overall twist, the helical shape, that's largely driven by water.
Okay, walk me through that.
Let's look at the components again. That sugar-phosphate backbone we just built, phosphate groups are loaded with negative charges. They are polar. They love water. They are hydrophilic.
So they naturally want to be on the outside of the molecule facing the watery environment of the cell's nucleus.
Exactly. They want to be solvated. But what about the bases? The A, T, C, and G, they are these flat, aromatic rings. They're relatively nonpolar. They don't have strong charges. They are hydrophobic.
So water-fearing, like oil.
Just like oil. So you have this chemical paradox built into one molecule. The backbone wants to dissolve in water, and the information-carrying bases want to hide from water.
So how does the molecule solve that problem?
It twists. The only way to satisfy both needs is for the hydrophilic backbone to form an outer shell, while the hydrophobic bases tuck themselves away into the core, away from the water.
And they stack on top of each other in the middle.
They stack like a roll of coins. This is driven by something called base stacking interactions, which are a form of van der Waals forces. By hiding in the middle and stacking together, they minimize their contact with water. The famous double helix is, in essence, a shape that keeps the bases dry.
That's incredible. So the helix isn't just some random, elegant shape. It's a direct consequence of its chemical parts being forced to exist in a water-based environment.
It is a structure born of and stabilized by its interaction with water. If you were to put DNA into, say, a nonpolar solvent like oil, it would adopt a very different shape.
All right, that's a perfect transition into the double helix itself. Section 3. We all know the names Watson and Crick, and we know Rosalind Franklin's crucial X-ray data showed them it was a helix. But there was another piece of the puzzle from a chemist named Erwin Chargaff.
Yes, Chargaff's rules. This was back in the late 1940s, and it was a monumental discovery. At the time, many scientists thought DNA was just a boring, repetitive polymer, maybe just ATCG over and over again.
A tetranucleotide.
Exactly. But Chargaff meticulously analyzed the base composition of DNA from many different species, from yeast to humans, and he found these strange, unwavering rules.
Which were?
No matter what organism he looked at, the amount of adenine was always almost exactly equal to the amount of thymine, and the amount of guanine was always equal to the amount of cytosine.
A equals T and G equals C.
Always. And the ratio of A plus T to G plus C could vary wildly between species, but that one-to-one pairing was constant. Watson and Crick saw this and realized it wasn't a coincidence. It had to be the key to the structure. A must physically pair with T, and G must physically pair with C.
And this pairing is held together by hydrogen bonds. It's like a chemical handshake between the bases on opposite strands.
It is. But, and this is a key detail that Harper emphasizes, not all handshakes are created equal.
What do you mean?
When adenine pairs with thymine, they form two hydrogen bonds. You can think of it as a relatively loose two-fingered handshake.
Yeah.
But when guanine pairs with cytosine, they fit together in a way that allows them to form three hydrogen bonds. That's a much firmer three-fingered grip.
Does that extra bond, that tighter grip, have a real biological consequence.
A massive one. It directly relates to the stability of the DNA. We talk about the melting temperature of a DNA molecule.
And by melting, we don't mean it turns to liquid.
No, in this context, melting or denaturation means the two strands of the helix separating, unzipping. If you heat DNA, the thermal energy will eventually overcome the hydrogen bonds holding the strands together.
And because the GC pair has that third bond.
It takes more energy, more heat, to break them apart. So a stretch of DNA that is rich in GC pairs has a much higher melting temperature than a stretch that is rich in AT pairs.
So here's a thought experiment based on that. If I'm an organism that lives in a boiling hot spring, like a thermophilic bacterium in Yellowstone,
Your genome had better be packed with G's and C's.
to keep it from falling apart in the heat.
Precisely. The GC content of an organism's genome is often correlated with the thermal environment it evolved in. It's an incredible example of molecular adaptation. And it's not just for bacteria in hot springs. In the lab, when we run a PCR test, like the ones used for COVID-19, we have to precisely control the temperature to melt the DNA.
And that temperature depends on the number of G's and C's in the target sequence.
If you get the temperature wrong, the DNA either doesn't melt or it melts too much, and the entire test fails. It's practical everyday chemistry.
Let's go back to the geometry for a moment.
Yeah.
We've established the 5' to 3' directionality of a single strand. But how do the two strands in the helix relate to each other?
They are antiparallel.
Meaning they run in opposite directions.
Yes. If one strand runs 5' to 3' going, say, top to bottom, its partner strand runs 5' to 3' going bottom to top. They're like two lanes of a highway with traffic flowing in opposite directions.
And that's necessary for the base pairing to work.
It's the only way it works. If you tried to line them up in parallel with both 5' ends at the top, the bases just wouldn't be in the right orientation to form those hydrogen bond handshakes. The geometry would be completely wrong.
Now, because of this twisting and pairing, Leninger points out that the helix isn't a perfectly smooth cylinder. It has these grooves.
The major and minor grooves, yes.
Yeah.
As the backbones twist around the central axis, they're not perfectly spaced. This creates a wide, deep groove, the major groove, and a narrower, shallower one, the minor groove.
And why are these grooves important?
They are access points. This gets back to your strict librarian analogy. The cell needs to read the information in the DNA, but constantly unzipping the entire helix is inefficient and dangerous. It exposes the fragile bases to damage.
So the grooves are a way to read the book without opening it.
That is the perfect way to describe it. Proteins, like transcription factors that need to find a specific gene to turn it on or off, can slide along the outside of the helix. They have chemical fingers that reach into the major groove.
And what do they feel in there?
The edges of the base pairs. The pattern of hydrogen bond donors and acceptors in the major groove is unique. For each base pair looks different from TA, which looks different from GC. So a protein can read the sequence by feeling the chemical signature on the outside of the helix, find its target sequence, and bind there, all without ever unzipping the DNA.
It's like reading Braille on the spine of a book.
Exactly like that. It's an incredibly elegant solution for information access.
There's one more chemical detail from Harper's that, well, it sounds a little scary. Tautomers.
It does sound a bit menacing, doesn't it? The attack of the tautomers.
It does. What are they?
It's a fundamental concept from chemistry. We draw the structures of the bases, A, T, C, G, as these nice static diagrams in textbooks. But in reality, at the quantum level, protons are not static. They can, for a fleeting moment, hop from one position on the molecule to another.
So the very shape of the letter changes.
For an instant, yes. An amino group can briefly become an imino group. A keto group can become an ino group. These different forms, which are in rapid equilibrium with each other, are called tautomers. It's like the letter A momentarily shape-shifting into something that looks a bit like a G.
And why is that a problem?
Because the entire system of base pairing, the A with T and G with C rule, relies on the bases being in their common, stable, tautomeric form. If a base just happens to be in one of its rare unstable forms at the exact millisecond that the DNA replication machinery is passing by,
The machine could misread it.
it could misread it. If an adenine is in its rare immunotautomeric form, its hydrogen bonding pattern suddenly looks like guanine, so the polymerase enzyme, following the rules, will incorrectly insert a cytosine opposite it instead of a thymine.
A typo. A mutation.
A spontaneous mutation. And this is the profound part. It's not caused by radiation or some external chemical, it's caused by the intrinsic, unavoidable quantum jitteriness of the atoms themselves. It implies that a certain low level of mutation is baked into the very physics of the system.
So even in a perfectly protected cell, the letters themselves can just wobble into the wrong shape and cause an error.
The letters wobble. And that wobble is a constant threat to the integrity of the archive. But as we'll see, it's also the ultimate source of all evolutionary novelty.
That's a great pivot point. We've built the library, we've examined the books and the paper they're printed on. But these nucleotides, they have other jobs, right? They aren't all just sitting in the DNA archive.
Oh, not at all. In fact, if you did a census of all the nucleotides in a cell at any given moment, the vast majority are not in DNA or RNA. They're out in the cytoplasm doing other things.
They're the cell's freelance workers.
They really are. And their most famous role is as the energy currency of the cell.
ATP, adenosine triphosphate. Harper's points out that the concentration of ATP inside a cell is huge, around 1 millimolar, which is orders of magnitude higher than other nucleotides.
It is the universal cache that every process in the cell accepts. But I want to clarify something about where the energy in ATP comes from. We often hear about high-energy phosphate bonds.
Right, which makes it sound like there's a little explosion when you break the bond.
It's a bit of a misnomer. It's not that the bond itself is strong. It's about the relief of repulsion. Imagine trying to hold 3 powerful, squirming magnets together, all with their north poles facing each other.
They'd be pushing apart like crazy. It would take a lot of force to hold them in a line.
That's ATP. You have 3 phosphate groups, each with a strong negative charge, all chemically bonded right next to each other. They are desperately trying to repel each other. The molecule is under immense electrostatic strain.
So it's a compressed spring.
A perfectly compressed spring. And when an enzyme cuts off that terminal third phosphate, the spring is released. That massive release of electrostatic repulsion is what provides the energy to drive other chemical reactions in the cell.
That makes so much more sense than a bond exploding. It's just relieving tension.
Precisely.
Now, one thing I've always been curious about, and Leninger speculates on this, why adenosine? Why is ATP the king? Why not GTP or CTP? They have the same phosphate spring.
It's a fantastic evolutionary question, and GTP is used as an energy source in specific processes like protein synthesis. But ATP is the universal standard. The leading hypothesis, as Leninger suggests, is that the adenosine part evolved as a sort of universal handle.
A handle.
Think of a socket wrench set. You have dozens of different sockets for different bolts, but they all click onto the same standard handle. In biochemistry, thousands of different enzymes that use nucleotide energy have a specific conserved protein structure called a Rossmann fold.
And that fold is perfectly shaped to grip onto the adenosine part of the molecule.
It fits like a hand in a glove. So the thinking is that once evolution designed this incredibly useful handle, it just kept reusing it for countless different tools. The phosphates do the work, but the adenosine is the part the enzyme grabs onto. It's an example of evolutionary lock-in.
So beyond energy, what else are they doing? Harper's highlights their role as second messengers.
Right, and here we see a crucial contrast in strategy. If ATP is the abundant cache in the economy, second messengers like cyclic AMP or CAMMP are the rare, powerful signals.
Harper says the concentration is in the nanomolar range. That's 1,000,000 times lower than ATP.
And that's the point. It's kept incredibly low on purpose. Think of it like a fire alarm. In a quiet library, even a soft bell is immediately noticeable.
But if the library were already full of loud bells, you'd need an air horn to get anyone's attention.
Exactly. Because the background level of TMP is near zero, when a hormone like adrenaline triggers a cell to produce even a tiny amount of it, the signal is massive and unambiguous. It shouts, "Emergency! Mobilize glucose now!"
And its cousin, CGMP, cyclic GMP.
That's another key signaling molecule, particularly famous for its role in smooth muscle relaxation and blood flow. The whole nitric oxide pathway, which is critical for regulating blood pressure, works through CGMP.
So drugs that affect blood vessel dilation.
Many of them, yes. A drug like nitroglycerin for angina or sildenafil for erectile dysfunction, they work by manipulating the levels of CGMP to cause blood vessels to relax and widen.
So they're the bricks, the money, the fire alarm, and they're also part of other tools, right? Coenzymes.
Yes, they're everywhere. Key metabolic players like coenzyme A, which is central to the citric acid cycle, or NAD plus AN, the electron carrier, they both have an adenosine nucleotide as a core part of their structure. Again, it's that useful handle. The cell just bolts it onto different tools to get different jobs done. They are the ultimate Swiss Army knife molecule.
Okay, this brings us to the point where this chemistry hits the bedside. Section 5, clinical relevance. Harper's has a great section on synthetic analogs, which sounds like we're building biochemical imposters.
That is exactly what we're doing. The strategy is often called anti-metabolite therapy, and it is a cornerstone of modern pharmacology, especially in cancer treatment. It's a Trojan horse strategy.
Walk me through it. How does the Trojan horse work here?
Okay, think about the defining characteristic of a cancer cell.
Uncontrolled, rapid division.
Exactly. It's growing and dividing furiously. And to do that, it needs to replicate its DNA, which means it is constantly gobbling up nucleotides A, T, C from its environment to build new DNA strands. It's greedy.
So we feed it something that looks like a nucleotide, but is secretly poison.
Precisely. We designed a fake nucleotide, a synthetic analog. It might look almost identical to, say, cytosine, but we've sneakily changed one atom. Maybe we've replaced a hydrogen with a fluorine or altered the sugar just a tiny bit.
The cancer cell, in its greedy haste, doesn't notice the difference.
It grabs our Trojan horse molecule and tries to incorporate it into its new strand of DNA.
And then what happens?
The whole process grinds to a halt. Either the fake nucleotide prevents the next one from being added, terminating the chain, or its presence distorts the helix so badly that the replication machinery gets stuck. The cell fails its pre-division quality control checks and is forced to undergo apoptosis-programmed cell death.
Can you give us an example of a specific drug that works this way?
Harper's mentions a classic one, cytarabine. It's a workhorse chemotherapy drug for certain leukemias and lymphomas. It's an analog of cytidine, which is cytosine plus a sugar.
So what's the flaw? What's the poison part?
The sugar is wrong. Instead of a normal ribose or deoxyribose, it has a sugar called arabinose. The only difference is the stereochemistry, the 3D arrangement of one hydroxyl group. It's an incredibly subtle change.
But it's enough to jam the machine.
It's enough. When cytarabine gets incorporated into DNA, that incorrectly oriented hydroxyl group physically blocks the next nucleotide from being added. Chain termination. The cancer cell can't replicate, so it dies.
What about another one mentioned, azathioprine?
Azathioprine is fascinating. It's a prodrug, meaning it's inactive when you take it. The body's metabolism converts it into a purine analog that gums up the works of both DNA and RNA synthesis. We use it not just in cancer, but heavily in organ transplantation.
To prevent the body from rejecting the new organ?
Exactly. And organ rejection is basically a massive immune response. Your T cells and B cells start dividing like crazy to attack what they see as a foreign invader. Azathioprine stops that. By feeding these rapidly dividing immune cells fake purines, we shut down their replication.
But this highlights the fundamental, brutal problem with this kind of therapy, doesn't it? The Trojan horse doesn't just fool the enemy soldiers.
No, it fools any cell that's dividing rapidly. That includes the cancer, yes. But it also includes your hair follicles, the lining of your gut, the stem cells in your bone marrow that make your blood cells.
Which is why chemotherapy causes hair loss, nausea, and immunosuppression.
It's the collateral damage. We are exploiting the fact that the tumor is dividing faster than most normal tissues, but it's still a very blunt instrument. We are poisoning the process of DNA synthesis itself and hoping the cancer dies before the patient does.
There's one more structural detail from Harper's in this section. Syn versus anticonformers. This felt pretty deep in the weeds of chemistry. Why is it clinically relevant?
It is deep in the weeds, but it's another beautiful example of how 3D shape is everything. Remember, the base is attached to the sugar by a single chemical bond.
So it can rotate.
It can rotate like a paddle on a stick. It can either swing away from the sugar, which is called the anticonformation, or it can swivel and swing back over the top of the sugar, which is the syn conformation.
Okay, like a door swinging open or closed.
A good analogy. Now, in our normal B-form DNA double helix, virtually all the bases are in the anticonformation. It's the most stable, least crowded position.
So why do we care about syn?
Because certain types of DNA damage or the incorporation of certain nucleotide analogs can force a base into the syn conformation. And when it's twisted into that awkward shape, it can't form the proper Watson-Crick hydrogen bonds. It disrupts the helix. It can cause a bulge or a kink in the DNA, which can stall replication or transcription. It's another way that a small change in geometry can have huge functional consequences.
It all comes back to shape.
Yeah.
Does the key fit the lock? If the key is bent into the wrong shape, the door won't open.
Biology is the science of shape.
I want to zoom out for our last section, to the long view. We started by talking about the stability of DNA, how the deoxy sugar makes it this incredible, robust archive. But Leninger makes a really profound philosophical point about this.
The Goldilocks stability?
Yes. Explain that.
Well, here's the paradox. For life to exist, you need DNA to be extremely stable. If your genetic code changed significantly from one day to the next, you wouldn't be a coherent organism. Your cells would lose their identity. It would be chaos, what we call an error catastrophe.
So stability is essential for an individual to survive. But
what if DNA were perfectly stable? What if the chemical bonds were unbreakable, if tautomers didn't exist, if no errors could ever be introduced?
nothing would ever change.
Exactly. No mutation means no genetic variation, and without variation, there's nothing for natural selection to act upon. Life would never have evolved beyond the first self-replicating molecule. We'd still be in the primordial soup.
So for life to evolve, the archive has to be just a little bit leaky.
It has to be in that Goldilocks zone. Stable enough to preserve information across the lifespan, but just unstable enough to allow for a slow, steady trickle of errors to generate novelty over evolutionary time.
And Leninger mentions specific slow reactions that happen, right?
Yeah.
This isn't just theory.
Right. Reactions like deamination, where a cytosine base can spontaneously lose an amino group and turn into a uracil, this happens thousands of times a day in every one of your cells.
And there are repair crews that fix it.
There are amazing, dedicated enzyme repair crews that are constantly patrolling your DNA, looking for these errors and fixing them, but they're not perfect. Every now and then, they miss one, and that misedit, if it occurs in a sperm or egg cell, becomes a permanent mutation passed on to the next generation.
So the very process that can cause genetic disease and cancer is also the fundamental engine of all evolution.
It is the ultimate trade-off.
Yeah.
You pay for the potential for evolutionary adaptation with the constant risk of genetic decay and disease.
And now, in our time, we've finally learned how to read this archive correctly.
It's been an incredible revolution. We started with Sanger sequencing, which was like reading the book one letter at a time with a tiny magnifying glass. It took years and billions of dollars to read the first human genome.
And now?
Now we have next-generation sequencing. The analogy is that we take 1,000 copies of the book, put them all through a paper shredder, and then dump all the shredded pieces onto a table.
That sounds like a mess.
It is. But we then use a super high-speed camera to read every tiny shred simultaneously. It's called massively parallel sequencing. And then we use powerful computers to look for overlapping phrases in the shreds, what we call contigs.
And the computer pieces the book back together from the shredded bits.
It reassembles the entire 3 billion letter text. We can now sequence a person's entire genome in about a day for less than the cost of a new television.
We've unlocked the library.
We have the full text. Now the real work begins. Figuring out what it all means.
So let's bring this all together. What's the final synthesis? We started with these simple 3-part bricks.
We saw how that sugar phosphate backbone created a hydrophilic shield with a specific 5' to 3' grammar. We saw how the hydrophobic bases stacked inside to hide from water, twisting the whole molecule into its iconic helix.
We saw the hydrogen bond handshakes 2 for AT, 3 for GC, that provide the specificity for copying the information.
And we learned it's not a static computer hard drive. It's a living, breathing, dynamic molecule. It's constantly being attacked by water. It's twisting, it's melting, it's being repaired and copied.
And beyond the archive, its component nucleotides are the money, ATP, the signal, CAMP, and the multipurpose tool, parts of coenzymes.
They are, without a doubt, the most versatile and important class of molecules in the body.
So here's a final thought for you to take away from this deep dive. For 4 billion years, the alphabet of life has had 4 letters. A, T, C, and G. That's it. But right now, synthetic biologists are creating new artificial nucleotides.
An expanded genetic alphabet.
Exactly. They've created base pairs they call X and Y that can be incorporated into DNA and replicated. What happens when we move beyond just reading the archive and we start writing our own chapters with a whole new alphabet? What kinds of new words, new proteins, new functions could we create?
We're on the cusp of moving from being readers of the book of life to being authors.
And that is both an incredible and a slightly terrifying thought.
A perfect place to end.
Thank you for joining us on this deep dive. Keep your backbones hydrophilic and your bases stacked.
Until next time.